Adrian Carter, Boehringer Ingelheim. Harnessing the creative power of the scientific crowd.
Good afternoon, everybody. I would like to talk in the space of the next 15 minutes to you about harnessing the creative power of the crowd and the importance of establishing a reliable framework for that. I will do that with a view from the pharmaceutical industry, unashamedly, and I'd like to start with a few home facts. First off, DeMasi published early this year that it costs $2.6 billion to turn a drug idea into a product. I also like to think about the rule of 10, that only 1 in 10 of the development candidates that start preclinical development actually get through to the clinic and are approved. And secondly, it takes about 10 years. So those two easy numbers you can recommend. It used to be a very linear process, basic research, discovery and development, and treatment. What we've seen over the last few years is a mixing, an intermixing between those defined groups. And one of the hopes we have in innovation healthcare is that academia will be able to provide us with many of the new ideas for drug targets. I hate to disappoint you, however, this may not be the case. This picture shows you the distribution, genes ranked by the number of publications in Gene to PubMed, and the number of publications per gene. These are only human Homo sapiens genes, 34,000 genes in that database as of a few days ago, 498,000 publications. That's the good news. The bad news is that 7,500 of those were for one particular gene, P53, the tumor suppressor. Even worse, over 8,000 of those genes had only one publication. So if we're expecting academia to be able to provide us with the ideas for this, we may be a little bit disappointed. Now you can also apply that exponential curve that you see. A mathematician will tell you that it looks like a power law distribution. That is, the probability of any particular event that you're measuring, in this case the publications, is inversely proportional to that number raised to its power of n. And you can do a log-log plot, and you can get roughly a straight line. Interesting, and this is really key for me, if you might not miss it, if you look at the top, the one that's marked OMIM is the online Mendelian inheritance map. This is telling you that there's a variety of different hits all the way across the entire spectrum of those genes. However, the gene which most people are working on is P53 now and was P53 10 years ago. So that is one of the biases we're facing when we're trying to find new targets in the healthcare world. It's also been given a name, the Matthew effect. From the Bible, for unto everyone that hath shall be given, and he shall have abundance. It actually refers to the fact that eminent scientists receive a disproportionate amount of recognition compared to junior scientists. There's actually some very good mathematical modeling that's gone here by Barabasi and Albert. They published about 10 years ago, and they use this to define scale-free networks. And a scale-free network is the sort of thing you see in the World Wide Web, but it's also what you see in academic groups. You see that they're growing continually, but they also demonstrate preferential attachment, which means that certain groups enjoy more recognition than other groups. And you can see what happens to that when you look at the gene distribution. The other big change that we have in the pharmaceutical environment, I think, has been that of open innovation. And I just wanted to spend a few moments to carefully define that term for you, because many people can understand different things about it. Originally, it was coined, and I apologize that the graph has really been thrown out. Originally, it was coined in 2003 by Henry Chesbrough, who used that to describe the term that external ideas as well as internal ones can be used to advance the R&D. Since that time, however, you've seen an explosion in different open terms. We have open source, which is something that's been pioneered by the software industry. Open source projects. We have open data. Open data is data that everyone can access and use. We have open access, which has revolutionized the publishing world. And we have open science, where people are pursuing their day-to-day science and publishing their lab books online, on time, at the end of every day. Now, I think there's a lot that we can learn in the pharmaceutical industry from the software industry. And the example I like to take is that of the new Linux project. In the 1980s, Richard Stallman developed the general purpose license, which allowed people to modify software and code and share it, with the goal of being able to find a replacement for Unix. Remember the IBM operating system. GNU is a recurring acronym, GNU. GNU is not Unix. This was extremely successful. In fact, most of your smartphones, Android smartphones, your desktop servers, your big computers for Twitter, Amazon, all the rest of it, run on Android-based systems. What the take-home message from much of that from the software industry were two things. It was that open innovation was important in defining the rules. It was also important that companies revealed certain things selectively in the hope that they in return would receive something of value. So in the second half of my talk now, I want to specify how one can go about selectively revealing in an open innovation environment, one built around crowdsourcing platforms, and the other built around public-private partnerships. Crowdsourcing platforms are a way of selectively revealing questions. You have to admit what you don't know and looking for potential solvers. And public-private partnerships in a truly open partnership are looking for ways of sharing tools amongst academics and industrial scientists. I shan't focus on bilateral collaborations. I'm sure you know more than enough about that. So, crowdsourcing. Again, start with the definition. Crowdsourcing is the act of taking a job, traditionally performed by a designated agent, fancy name for an employee, and outsourcing it to an undefined, generally large group of people in the form of an open call. I like the soundbite version, the application of open source principles to fields outside of software. And I'd like to share two examples with you, one BioMedX, based in Heidelberg, and the other is the Innocentive, the crowdsourcing platform. Crowdsourcing with Innocentive. It basically means that you formulate a challenge by a so-called seeker, suggest problems, formulate those into challenges, post those challenges in the appropriate channels, evaluate the solutions coming out of it, and then, after evaluating, reward those ones that you want to reward and you want to take on board. We've gone through the process ourselves 12 times now, 2,169 registrations, 361 solutions, 33 awards, 16 potential collaborations. Sounds great, doesn't it? Unfortunately, the last bit is the difficult one, 16. It's when you get into trouble with tech transfer organizations, because then you have to organize the agreement which sets you up to be able to work together with them. The BioMedX model, a company based in Heidelberg, a startup, has elements of crowdsourcing in it, worldwide call for project proposals, crowdsort those proposals are prioritized and selected. However, the fundamental difference here is that not only are the solutions selected, but also the young scientists with those solutions. And you bring them together in a boot camp, and you sort them out, and you pick the best ones, not necessarily the best individuals, but the ones that work best with each other. And you form the nucleus of a team around them, and you set them up in a facility. And you give them the opportunity in order to be able to turn their creative idea into something meaningful. This is one team we've been working with, a team of individuals. On the right of the picture, 399 registered solvers, total from 59 countries, full project proposals around about 170. You can see the same sort of process here as we had with Innocentive. Internally evaluated 99, we invited 23 to the boot camp, and the result is what you can see on the right-hand side, those individuals. They're actually working here and exploring the epigenetic regulation and development and progression of COPD. The last example is that of a public-private partnership, and I would like to use the example of the Structural Genomics Consortium, SGC. I think you heard a little bit earlier on today about the importance of epigenetics. Well, this is a consortium of 8 pharma companies, Boehringer Ingelheim being one of them, which sit together with academic institutes based in Toronto, Oxford, in Zurich, in Karolinska, and in Campinas in Brazil. And the idea behind it is to develop chemical tools together by understanding the protein structure, which then allows them, make those public, which then allows everybody to be able to access the results coming out of it and be able to turn it into their own industrial project. So we have what I call a Creative Commons area, which is open, no restriction, no reach-through, no patents, but at some point you can then run with that particular idea and turn that into a potential drug. Now, the idea behind this, and you can see on the left-hand slide, a distribution of citations for nuclear hormone receptors. Nuclear hormone receptors are receptors which control gene transcription on the inside of cells. The blue ones are those that have been ranked according to citations in which proteins have chemical probes available. The red ones down at the bottom and the tail end of our exponential distribution are the ones which don't have chemical probes. The fundamental thesis behind this is that if you provide academic scientists with the chemical tools in which they can explore certain biological effects, you get to learn more about those proteins. It's rather like putting a saucer of milk down. It encourages people to work in that particular area. Now, that could sound very one-sided from the pharmaceutical industry. They were just taking, they're not giving. But the example on the right-hand side is an example that we had earlier on this year. We were interested in the bromodomain 9s, which is a reader protein, an epigenetic reader protein. And we, together with the SGC, have made a chemical probe available. You can find that on the chemicalprobes.org website. You can go there. You can click on the site. You can download it. You can buy it. And you can use it without any strings attached, because we want to encourage people, much as other companies have done and other chemical probes, to use those chemical probes in order to be able to understand the biology a lot better. You can also apply this to other areas, so there's a strong interest in the kinases. You have over 500 kinases, which I can tell you give you the same distribution if you look at the publications. Many academic scientists are focusing on a few of the kinases, whereas what the biology, the GWAS, and the OMIM studies are telling us is that there's a lot more activity, genetic interest in the other kinases, which are relatively unexplored. And perhaps together in a public-private partnership, one can characterize and make chemical probes available for companies and academics to be able to work more on them. Don't just take my word for it. I would highly recommend you dig into a supplement which we sponsored earlier on this year in May, an entire Nature supplement all about open innovation. It has chapters on open innovation, on the chemical probes which I've just talked about, compound screening, how the scientific process is being opened in different ways, the challenges of crowdsourcing solutions, and also the way many of these are being applied to areas which wouldn't normally generate a great deal of interest for the large pharmaceutical companies, unfortunately, such as the areas of tropical disease, where they're finding new ways of working together between academia, small startups, and big pharmaceutical companies and open partnerships. And on that note, ladies and gentlemen, I would hope that you'll be able to listen to the speakers after me as much as you've just listened to me, and I hopefully they'll be able to finish on time. Thank you very much.